Hello everyone, good evening. Am I audible to you all? Yes, please join the class fast and confirm if I'm audible to you or not. Let's wait for other students to join the class and we'll start the class at sharp 8 p.m. Okay? Good evening everyone. Markers today, okay? We are left with two markers. First is RAPT and then AFLP, okay? We have already covered RFLP in the previous class and the introduction to molecular marker. So you can go back to our live session of previous class and watch that. And I would suggest all of you to join the session at 8 p.m. sharp because we'll be starting with the class. We won't wait for other students to join. Okay, so let's start the class. First of all, welcome you all. Good evening, everyone. And this is our extended class of molecular marker. Okay, this is our full course which is running and the crash course which is running in our app. Okay, we have an application. You can download it from Play Store and iOS as well. Also, you can order our notes booklet from Botany Insider application itself. Also, for all the updates of the class, for all the updates of this marathon class, we have a WhatsApp community group. Okay, WhatsApp community group. So you all can join there and get the update of the class on the daily basis. For all the links you will find in the description box. Okay, let's start with the class. This is the second class of molecular marker. We will be dealing with first RAPD and then AFLP. We have already dealt with RFLP in the previous class. So, and also we have a PYQ course which will be launching very soon on our Botany Insider app. So make sure you enroll in this batch also. Make sure you join in this batch which will be launching very soon on our Botany Insider application. And here you will be practicing last 10-year questions, okay? From 2023 to 2013, okay? So today's session is about the RAPD and AFLP marker. What is the full form of RAPD? It is random amplified polymorphic DNA. It is strictly based on PCR method. Okay. The other marker which we study, RFLP, was strictly based on restriction digestion basis. Okay. So from the name of it, it suggests that it is random amplified polymorphic DNA. In case of random amplified polymorphic DNA, first of all, what polymorphic means, in the previous class also, I've told you that the DNA portion in one organism and in the next organism is going to be different. In two individual or two different species, the DNA sequences are going to differ. This is known as polymorphic DNA. And it is saying that it is randomly amplified DNA. So what we are doing is that we first have DNA sequences of two organisms. This is the first organism and this is the second organism. In this method, we use 8 to 12 base pair primer sequences for PCR. Okay. Why we are using 8 to 12 base pair primer sequences? Because this is the ideal size of primer. Because if the primer is going to be too long, then it will become too specific and it will not bind to many places in the DNA sequence. But if it will be too small, then it will bind to almost all the places in the DNA sequence and it will give us many large fragments which we don't want. So we use 8 to 12 base pair primer sequence which is the ideal size for PCR reaction. Okay, and we are using random primer sequence because the primer sequence are not specific here. So they are going to bind at any location. Okay, for example, in this DNA sequence, it is binding at two places, and in this DNA sequence, it is only binding at this place. Okay. So in the first case, we will be getting two fragments, and in the second case, we will be getting one fragment only. Now, what we do is we run gel electrophoresis for this. What happen in gel electrophoresis is that we have DNA wells. Okay. We load first organism's DNA here and second here. So in this case, we are getting two bands, and in the second case, we are getting only one band because there is only one sequence here. Okay, one fragment which is obtained here. So what we do is now we study these sequences and visualize it in autoradiograph. Now we use these DNA sequences for various type of studies such as gene mapping to identify different species. Okay, and also in phylogenetic analysis. Okay, is this clear to you all? Yes, please confirm if this is clear to you all. This is the most basic method which can be used in molecular marker techniques because it do not require any specific primer and we do not need to have a prior knowledge of the DNA sequence. Okay, this is the major advantage of using this method that we do not need a prior DNA sequence knowledge. Okay, is this far clear to you all? Yes. Please check if anyone have any doubt. And if you haven't watched the first molecular marker, which I've already told you in the previous class, I would suggest that you also watch this class. Okay. In case of RAPD, it is random amplified polymorphic DNA. It is a molecular biology technique which is used to generate DNA fingerprints or profiles of individual organism and species. It is a PCR based method. It is the most important point here that it is a PCR based method means it is a polymerase chain reaction based method which involves the use of short random DNA primers, which is used to amplify segments of genomic DNA. And these primers are typically of 10 to 20 nucleotide long, which are chosen arbitrarily. They are not specific. Okay. And this leads to amplification of a random subset of DNA fragment. What we do in this technique that we first isolate our DNA fragment from the genomic DNA. We extract the genome of our interest and then we do the PCR amplification step. We use short single stranded DNA primer which is used in PCR reaction with the genomic DNA template. And these primers are typically short sequences so that they can bind to multiple locations in the genome due to their arbitrary nature. When the primer anneal to complementary sequence in DNA polymerase extends the primer and result in amplification of the DNA. From one fragment, it gives two fragment and from two further, it amplifies our DNA. So even if we have very small amount of DNA, it can amplify it and show us the result. Okay. Then we perform gel electrophoresis. What we do in gel electrophoresis is that it separates the molecules on the basis of their size. Okay. And this technique uses electric field. In running gel electrophoresis, what we do is we run fragment from negative direction to positive direction because we know that our DNA is negatively charged. So it is going to move from negative point to positive direction. Okay, on the gel matrix. Shorter the fragment, it moves the faster, and the longer the fragment moves slower. Then we do the visualization and our required study. Then these DNA fragments are visualized and using staining, and they are radioactively labeled if it is required. Okay. This is the technique that we have plant sample 1 or sample 2. What we do first is we isolate the DNA sequence that we want to amplify or we want to study. Then we do the random primer annealing. Okay. The primer are going to anneal at the random locations in the DNA sequence. Then we run gel electrophoresis and we do the visualization. Okay. Now, what are the advantages or disadvantages of using this technique? The first and major advantages of using this technique is that it do not require prior knowledge of DNA sequence because we are using non-specific primers. And it can amplify even the smallest amount of DNA. Okay. It has simplicity to it because the procedure, when compared to RFLP and AFLP, is relatively easier and it is cost effective also. The RAPD is relatively simpler and does not require the prior knowledge of genome or the need to design specific primer for the specific loci. This made it the cost effective method also. Okay. It requires minimal DNA because it is later going to amplify that DNA. And it do not require the prior information of the DNA sequence. RAPD does not require prior sequence information about the genome being studied. This can be advantageous when working with organisms for which genome information is listed or unavailable. Okay, and it also gives the rapid result and it also identifies the genetic diversity. It can help in identifying genetic diversity within or between population, making it useful in study of population in genetics and evolutionary biology. I hope this far RAPD is clear to you all. Yes, please confirm if anyone have any doubt. Please check if this procedure is clear to you all. Are you still facing the voice issue or is it GM 1.5 speed? Yes, others please check if my voice is breaking or something. Am I properly audible to you all? Yes. Can you all confirm if the voice is clear to you or not? Okay, okay. Thank you so much. Okay. Now, these are the disadvantages of using this method. First, it is less specific. Okay, because we are not using any specific primer here. Okay, so the primer is going to be bind anywhere in the DNA sequence and it is going to give us arbitrary result. And the only disadvantage other than this is that it is not reproducible. Okay, because since the primer is not specific, it becomes very difficult to reproduce these results. Okay, so there is a lack of reproducibility. RFID results can be sensitive, but making the technique less reproducible compared to other DNA fingerprinting method. Okay, and we are using dominant marker. Okay, it cannot differentiate between heterozygous and homozygous. Okay, so it is a dominant type of marker and it provides limited information, provides information about the presence or absence of a particular DNA fragment, but it does not provide information about the specific location of that fragment. Okay, and there is no sequence information. It does not offer sequence data. Other than this, there is difficulty in comparison. Okay, comparing RAPD profile between different laboratories can be challenging because there is a lack of standardized primers and condition. This makes it hard to share and compare data across studies. It is also susceptible to DNA contamination because it is leading to the false positive results also, and there is limited resolving power. It is very hard to interpret these results as well. Okay. These are the RAPD markers. Okay, they are using the PCR method in which arbitrary primed PCR we are using, and it is used for DNA amplification fingerprinting. First, what we do is we run the template and we use non-specific primers in this case. These primers are going to anneal at the non-specific locations and then these are going to result into DNA fragment being generated. Okay. Now the other method we use is AFLP method. Okay. AFLP is amplified fragment length polymorphism. This is the advanced method or the combination of methods in which we use both the techniques of RFLP and RAPD. In this case, we are using restriction digestion that we used in RFLP, and we are also using PCR technique that we used in RAPD. Okay, so this is the most advanced method that we use. What we do is that we have a DNA sequence. Okay, first what we do is that we restrict it using digest it using restriction enzyme. Then it is going to generate fragments. Then we run the PCR technique and amplify this method. Okay. Now, what we do is, first let me select the panel. Okay. Now, in the case of AFLP marker, what we first do is that we have a DNA sequence. Okay. Here we have a site for, let's say, MSV1 restriction enzyme, and here we have site for EcoR1 restriction enzyme. These are the type of restriction enzyme which are going to recognize their specific recognition sites and cut the DNA at their specific locus. For EcoR1, we have GAATC, GAAA, and here we have, okay. For suppose it is cutting here. Okay. So what we have is now we have generation of sticky ends. These type of ends are known as sticky ends. Now what we do is we add adapters to it. Okay. To run the PCR reaction. Okay, these adapters are going to be specific to these adapters are going to be complementary to our EcoR1 or MSV1 site. Okay, these adapters are going to bind with the complementary part of the restriction site present in EcoR1 and MSV1. Then we have generated a blunt end. Then what we do is now we run the PCR cycle and we do gel electrophoresis. We consider here only the DNA fragment of one organism. Now suppose we are doing it for another organism as well. And in the other organism, for suppose there are two sites for EcoR1 and one site for MSV1. So it is going to generate two fragments, right? Now we run the gel electrophoresis for this. This is for organism 1 and this is for organism 2. Here we are getting one fragment and here we are getting two fragment. Now we do is we study this and visualize it. Okay. The major advantage or the major focus of this technique is that we are using specific EcoR1 or specific restriction sites first to digest this, and when we run the PCR cycle, we use specific primers for the amplification. Okay, because these primers are going to be specific for our adapter sequence. So even if we don't know the sequence of the DNA that we want to amplify, we can still amplify because we know the sequence of the adapter which is complementary to our EcoR1 and we easily know the restriction sites. Okay, so by knowing the restriction site, we know the adapter site, and by knowing the adapter site, we can design our specific primers. Okay, and these specific primers provide this technique the specificity and also makes this technique very reproducible. Okay. In case of AFLP, what we have is amplified fragment length polymorphism because we are getting different lengths of DNA and we are amplifying that DNA sequence. Okay. It is a molecular biology technique which is used in DNA fingerprinting and genetic analysis of organism. Like RAPD, AFLP is PCR based and it is used to detect genetic variation among individuals. How this work is, we first digest our DNA using restriction enzyme and that is going to cut the DNA at specific recognition site, generating a collection of DNA fragments of varies length. Okay, then we are going to ligate it with the pre-selective adapters. Okay, these are adapters going to be complementary to our restriction site. Complementary to restriction site. These are going to ligate to the end of restriction fragment and these adapter contain sequence that are recognized by specific primer used in later step. These specific primers now going to be complementary to our adapter sequence. And we do the selective amplification technique. In case of selective amplification, we need a set of primers which are used to target the adapter sequence and specific selective bases. These are added to 3' end of the primer and it is going to be complementary to the nucleotide adjacent to restriction site. Then we run our gel electrophoresis and we visualize the DNA. This is the technique in which here it is using PST1 and EcoR1 as the restriction enzyme and these are going to cut the DNA at the specific location. Now we use the adapter. The first adapter is going to be specific for EcoR1 and the other adapter is going to be specific for PST1. Then they do is they generate the blunt end. Now we design primers, okay, which are going to be complementary to our adapter sequence. Now we do the amplification step, and after amplification we run the gel electrophoresis and do our required study. Okay, is the method AFLP clear? This is the most advanced method in molecular markers. Okay, because it combines the knowledge of RFLP and RAPD. And this is the major marker we use in DNA fingerprinting. Okay, in case of phylogenetic analysis, in case of species recognition. Is this clear? Yes, confirm everyone. Others, check if it is clear to you all. Okay. So, the major advantages of using AFLP is that it provide high reproducibility. It is generally more reproducible than RAPD as it involves a specific adapter sequence and selective basis. It is a dominant and co-dominant marker, means it can differentiate between heterozygous and homozygous. It can be treated as a dominant or the codominant marker depending on the type of analysis we want to perform. And it provides the intermediate level of resolution. It provides a higher level of resolution than RAPD, allowing the detection of more genetic variation among the individual. Also, it is applicable to various organisms, to a wide range of organisms, including plants, animals, and microorganisms. The major disadvantages of using AFLP is that it is labor intensive because the process includes many steps. First, we digest the DNA, then we are ligating the adapter, then selective amplification. So it makes the process tedious, and it is also cost efficient. And no sequence information is required in this step. Like RAPD, AFLP marker do not provide any sequence information about the amplified fragment, and it also provides the limited multiplexing. Okay, are the markers all clear to you all? We are done with all the three markers RFLP, RAPD, and AFLP. Okay, first marker was restriction digestion based or hybridization based. Other two markers were PCR based. This one was restriction digestion plus PCR. Okay. Yes, confirm if the methods are clear to you. We are done with all the molecular marker. Now in the next class, we will deal with all the techniques of gene knockout and gene knock-in. Okay. From Monday onwards at sharp 8 p.m. in the marathon class. Okay. This was all about the marker. And additionally, there are two courses running, full course and crash course. You can enroll for the preparation of December 2023 because there is not much time left in the exam. So make sure that you are completing your syllabus and revising it properly. Also, if you are feeling any lag in your preparation, you can purchase our notes booklet. And for PYQ course, we will be launching this course very soon. So you can enroll in this batch. Okay. Can you please explain a bit about how dominant and cold are? Okay. Bye bye everyone.